Welcome. Today we are going to discuss on recombinant vaccines. Let's see what vaccines are. Vaccines are preparation of killed or weakened microorganism that is given to a person orally or injected in order to prevent diseases. In 1798, Edward Jenner was the first person to immunize people against smallpox using cowpox virus. Later come killed and attenuated polio vaccines, and kill vaccine was used by Jonas Salk and live vaccine by Albert Sabin. Coming to the properties of a good vaccine, a vaccine should have the ability to elicit appropriate immune response for a particular pathogen. It should give a long-term protection, safety, stability, and it should be inexpensive. Types of vaccines. There are different types of vaccines: live vaccines, killed or whole organism vaccine, subunit vaccines, recombinant vaccines, and DNA vaccines. Let us concentrate our talk in recombinant vaccine. By using recombinant DNA technology, the genes encoding any immunogenic protein can be cloned and expressed in bacterial, yeast, and mammalian cells. The first such recombinant antigen vaccine approved for human use is hepatitis B vaccine. This vaccine was developed by cloning the gene for the major surface antigen of herpes B virus and expressing it in yeast cells. Types of recombinant vaccines. The recombinant vaccines may be broadly categorized into three groups. First one, subunit recombinant vaccines. These are the components of the pathogenic organisms. Subunit vaccines include proteins, peptides, and DNA. Hepatitis B virus antigen comes under subunit recombinant vaccines. Next is attenuated recombinant vaccines. These are the genetically modified pathogenic organisms that are made non-pathogenic and used as vaccines. That is, in bacteria or viruses, the virulence causing region is removed and they are made non-pathogenic. The third one is vector recombinant vaccines. These are the genetically modified viral vectors that can be used as vaccines against pathogens. So, in order to use a vector in recombinant DNA technology, there should be some properties. It should satisfy the properties. The major one is it must be compatible with the host system, and it needs a good combination of strong promoters, ribosome binding sites, termination sequences, affinity tag, and it should contain multi-enzyme restriction site. In the last classes, we have seen the tools for genetic engineering and also the rDNA technology protocol. So I am going straight for the preparation of a genetically engineered vaccine. That is, here we can see a hepatitis B virus, and this is a genome, and we are isolating a surface antigen gene. This is the surface antigen gene we have isolated. Now we have to insert it into a vector, and we are inserting it into the yeast vector. Now our yeast cells have become transformed yeast cells. We are purifying the hepatitis B antigen and we are using it as vaccine. This is a flow chart showing the preparation of a genetically engineered vaccine. Now we will go in detail. That is, here we can see the cloned HBV DNA from where we are isolating the hepatitis B virus and antigen. Then we are inserting it into the yeast DNA vector by ligation. Now these yeast cells have become the transformed yeast cells. These vectors are transformed into yeast cells and we will get transformed yeast cells. These transformed yeast cells are grown on medium selective for plasmid containing cells. Then we are culturing the cells and we are isolating the cells by centrifugation, lysing the yeast cells, and we isolate the protein. That is, here we are isolating hepatitis surface antigen protein. Now coming to the advantages of the recombinant vaccine. Produce more quickly in larger quantities, free from infectious virus particles. They can safely be given to immunosuppressed people, and they are less likely to induce side effects. Disadvantages: antigens may not retain their native conformation, so that antibodies produced against the subunit may not recognize the same protein on the pathogen surface. Isolated protein does not stimulate the immune system as well as whole organism vaccine. These are my references, and